saliva. And what's in saliva? Amylase. Correct. So it's the saliva that joins the mechanical chewing and everything, and therefore you have both taking place. Right, guys, let's move it. Let's move on. We're going to start with the digestive system. Amylase is an enzyme released by the salivary glands, and it breaks down starch into glucose. Guys, can I ask one random question? Is everyone up for it? An absolute random question that has nothing to do with what we're doing here, but we have covered it in the last few weeks. You know, through the phloem, right? We know that the phloem transports food, but what are the two molecules that it actually transports? What are the specific names? What are the specific molecules that it transfers? And while you guys answer that, I'm going to find out who's late, because some people have not joined the lesson yet, and we're 7 minutes in. What is this? I kind of gave you a mega hint there. I gave you a mega hint. It's not water. Water is through the xylem. Oh, yes, man, you're almost correct. There's two molecules which have been broken down. They're smaller now. Tarek, you're thinking the right thing. I'm thinking of something smaller, though. Thank you, Annie. That's fine. Just made a note. Nathan locked in. Oh, hold on. You're missing the other one. Well done, Zia. Anyone else? That's the one, Yusma. That's the one. Oh, wait, hold on. What's the scientific name for the first one you mentioned? The scientific name for the first one you mentioned. Correct. Well done. So you could have said amino acids and glucose. You could have said amino acids and sugar. Okay. I think if you said sucrose, you might get away with it. I need to look into that one. But it is amino acids and, because proteins is a big molecule. Our protein's gonna fit through the sieve plates. Our protein's gonna fit through the sieve plates. Does that make sense? Like, as in, do you see why that you can't write that? Okay, right, guys. I'm just trying to find out where all the latecomers are. Then we shall begin. Bada bing, bada boom. Okay, right. Bum, bum, bum. Okay, so let's begin, people. So we're talking about the digestive system. Everyone remembers this picture. We spoke about how the mouth is the mechanical movement. Salivary glands have the enzymes such as amylase. Then it moves down the esophagus. Guys, the lining of the esophagus is covered in smooth muscle, right? So the lining of the esophagus is covered in smooth muscle. The smooth muscle allows for what process? Begins with a P. You can't hear me. We did end at liver, that's correct. We're just going to speed through the first few. Very good. Well done, Sia. Well done, Nathan. Good. So that process was called peristalsis. Okay, so that muscular movement is like pinching the esophagus, forcing the food to go down. Is that okay, guys? Then we get into the stomach, and in the stomach, food meets HCl, so really low pH acid. So really low pH acid. Is smooth muscle similar to soft tissue in arms? Ooh, no, not quite. In arms, you'll get skeletal muscles, and hold on. Can everyone hear me? By the way, I'm getting some people saying they can't hear me. Can you hear me? Sorry, guys. We're due to have started ages ago, but technical difficulties is causing this to be delayed. We will make up for lost time, though. We will make up for lost time. Thank you, Manya. Thank you, Annie. Awesome. Okay, thank you, Sarah. All right, I think that's a consensus there. You guys can hear me. Yeah, you did. We're just doing a quick recap. So the stomach has low pH, okay, which is that HCl, but there's also mechanical movement in the stomach. Right? Food is churned. Does this all ring a bell, yeah? Food is churned. Right, okay. Then we started talking about the differences. Why are some structures labeled with a blue? Why are some structures labeled with a red? And that's because... Why is that the case, guys? Because the stomach is where the food actually passes through. Okay? And yes, that's correct. The red ones is where there's chemical involvement. Okay? Awesome. Let's move on. So we're talking about the stomach. We said there's churning, there's muscular movement, and the next one is protease. So protein is broken down in the stomach. So protease meets proteins and is broken down in the stomach. And the next thing we just spoke about was HCl. Guys, what else does HCl do? So HCl does what? It breaks down food. What else does it do? Very good. It destroys pathogens. Okay, we spoke about this. Okay, 1 second. Okay. Right. So where we are now, we are at... we're talking liver now. So we said that liver produces a lot of bile, right? What does the bile do? It neutralizes stomach acid, right? So we said that the liver produces bile. We're moving on, people. And emulsifies fats. Well done. So emulsifies fats. What does emulsification of fats mean? Emulsification of fats means taking a large fat structure and breaking it down into smaller fat structures. So smaller globules. Correct, yeah. So they increase the surface area so that enzymes can get to work. What enzymes are we referring to? So it increases surface area so enzymes can get to work. Well done. Thank you, Rahul. Yes, that's right, Manya. And the enzyme is lipase. Well done, guys, for saying lipase. Yes, we didn't finish it, Ali, so that's why we're just doing a quick recap and we're gonna continue now. So we're going on to new stuff now. So we're gonna talk about the gallbladder. So what happens in the gallbladder? So this is where bile is stored. Okay? This is where bile is stored. Where is bile made? Bile is made in the liver, but it's stored in the bladder. You know your actual bladder, what does it do? What does your actual bladder do? Everyone, please let me know. Stores urine, right? So a bladder is for storage. It doesn't produce urine. It stores urine. So well done, guys. So what does the gallbladder do? It stores bile. Okay. Awesome. Let's move on. Is there anything here at the bile duct? Let's see if I've got the bile duct. So it's released into the duodenum. I don't know how to spell that word. Pronounce that word via the bile duct. So it travels through here. Okay, let's go. Let's move on. We're going on to the pancreas now, guys. Now, what did we say that the pancreas produces? What does the pancreas produce in terms of digestion? Not in terms of blood being broken, blood sugar being broken down. What does it release in terms of digestion, guys? What does it say for esophagus? Peristalsis. So I said, what does the pancreas do for digestion? So Nathan, that's one of them. Well done. Adam, you can hear me now, right? So that's for the breakdown of sugar. I'm putting that blood sugar is completely to the side, guys. You know blood sugar, yeah. What what process is that involved in? It begins with forgotten the word. It begins with, oh my days, how have I forgotten this word? So osmoregulation, thermoregulation, and blood sugar. What's the word? That makes everything stay at homeostasis. That's the one. Thank you, Adam. Right. So sugar is for homeostasis, right? That's a negative feedback, correct? But for digestion, there's 3 enzymes released by the pancreas, and that is carbohydrates, protease, and lipase. So it actually releases a lot of enzymes. Carbohydrates is more of a generic name. Amylase is a bit more specific. Okay. Let's move on, people. We're going on to the small intestine. Yeah, they're the same thing. Correct. At this stage, they're the same thing. What travels through the bile duct? What travels through the bile duct is bile. Okay, small intestine. So, what happens in the small intestine? So, remember the contents of the stomach are what? Well, you know when the contents of the stomach are mixed in with the HCl, what happens, guys? So remember, the stomach connects to the small intestine. The stomach connects to the small intestine. So the contents of the stomach is acidic. Would you agree? The contents of the stomach is acidic. So in the small intestine, that's where it meets bile. Okay? Correct. That's right, Tarek. Food nutrients. Correct. Yeah, that's right. So in the small intestine, right, the acid is neutralized by bile. Bile is what? Slightly alkaline. So it neutralizes it. So it means that you're not gonna, have this extremely acidic small intestine. Right? This is where enzymes get to work. So small intestine, enzymes get to work. Now essentially what happens here? Large food molecules are broken down into small food molecules, and they are absorbed through the microvilli. They're absorbed through the walls of the small intestine. I'm running out of space here, guys. They're absorbed through the wall of small intestine, through microvilli, okay, into the what? Into blood. Do you have to know this word for word? Yes. So I'll say it again. In the small intestine, enzymes get to work, right? What enzymes are working in the small intestine, guys? Lipase is turning lipids into glycerol and fatty acids, right? Is that correct? Is that correct? Lipids broken down into glycerol and fatty acids. Proteins are converting, sorry, protease is converting proteins into, finish that sentence for me, please. Thank you, Nathan. Thank you, Manuel. Protease is turning proteins into, well done, Adam, well done, Yosma, amino acids. Okay, awesome. And the last one is amylase is converting starch into glucose, right? So those big molecules, starch is too big. It can't move through the walls of the small intestine. Okay? Proteins is too big. It can't move through the walls of the small intestine. And lastly, lipids are too big. They can't move through the walls of the small intestine. So these enzymes make them small enough so that it can be put into our blood. Does that make sense? Awesome. Okay, so we've done that. Let's go to the large intestine. So the large intestine is where remaining water is reabsorbed. Okay, and what's left behind? The number 2. Okay, so water is reabsorbed. Okay, water is reabsorbed, leaving a waste product called feces. Okay, and then of course the feces, they move through the rectum. Okay, and then they are ejected, or egestion is taking place, where they leave through the anus. Egestion just means removed or rejected. What do you mean? What do you mean? So water is being removed at every stage. At the large intestine, it's the last step. Any remaining bits of water is removed. I think that's everything, guys. A few more bits about the large intestine. Sorry, guys, I'm moving slow today and less emphatic because I'm just fighting this demon, and by that I mean this fever. Did it go through the stomach, or did what go through the stomach? Parts of the water. It's okay, uncle. Parts of the water. A lot of the water comes out in the small intestine as well. I know, I have no space. You know what, Tarek? I was reading an article about the appendix. A lot of people for a long time thought the appendix had no use up until like a month ago, where there was a big discovery about the role of the appendix. So and you know why that's an interesting one, because a lot of people who believe in evolution believe that the appendix is an evidence of evolution because we have run out of a use for it. But actually, there is a use for it. We'll get into it. Not right now, though. And it's something to do with the microbiome of the stomach. I mean, of the large intestine. I'm going to be honest with you. I don't remember all the details. That's why I don't want to get into it right now. There'll be a time and a place. Right. Okay, guys. We're moving on. What does it have? What does it do? I don't. Right. Obviously, a lot of people get it removed. It doesn't mean they're now deficient. It does have a very subtle. It's a very subtle use. We don't need to know about the appendix. Okay, sure. Yes, it's about gut bacteria, correct. But they found like more stuff about it. Right, guys, can I move on? So this is where we are. Can I move on? Let me know in the chat. Somewhat, Ali, somewhat. I can't believe it though, because we've got so much to cover. Awesome. Right, let's move on, people. Let's move on. The importance of bacteria in digestion. So the large intestine is home to hundreds of species of bacteria. You might have heard of gut microbiome before. Anyone here have Yakult and what's the other thing? There's a lot of foods actually that people have, like fermented foods, like yogurt and stuff, which really helps with the microbiome. These bacteria form the microbial ecosystem that play an essential role in human digestion of food. One, by breaking down substances we can't digest, supplying essential nutrients, and providing competition with any harmful bacteria. So those bacteria, yeah, that's it exactly. And kimchi as well. Apparently that's a good one. Actimel is definitely one. But yeah, so these things, they can kind of enhance your gut bacteria, and essentially those bacteria in your gut, they do further breaking down. They supply essential nutrients because when they break things down, they release nutrients, right? They don't kill bacteria. They are yeah, I guess they do actually kill bacteria. They cause them to die because they're eating their food, and they're providing competition with any harmful bacteria. Everyone knows what competition is, right? So they'll compete. They'll compete for space. They'll compete for nutrients. They'll compete for resources. That kind of stuff. So because of that, what happens to the harmful bacteria? They die. So really, what do you want in your stomach? You want a high. Who can finish that word? That's the one, man. Yeah. Ali, man. Is that a typo? Let me know if that's a typo. So yeah, thank you, Nathan. Thank you. So we want a height. Did I write height? I'm gonna put this down to my to my illness. Biodiversity. Biodiversity. So that's why you should have a range of fruit, a range of veg, a range of everything, because you're going to expose your gut to multiple different harmless bacteria, or should I say good bacteria, good gut bacteria. Let me know if that makes sense, people. We're moving on, yeah? We're moving on. Okay, so where we at? Okay, enzymes. Does everyone know what lock and key theory is? Okay, there's some stuff here that I'm gonna have to skip because they're just too easy, and I know you guys know it really well. So basically, enzymes, they have a specific shape, don't they? They have a specific shape, which means they're specific to... Can anyone finish that sentence? Enzymes have a specific shape, which means they are specific to a particular... Begins with an S. Correct. They are... Good. Well done. They are specific to a particular substrate, which means that amylase will only deal with carbohydrates, right? Exactly. And that lipase will only work with lipids, and that what's it called? Protease will only work with proteins. Does that make sense? So they're very specific, which means they can be used to control. It's like clockwork. These enzymes are like clockwork because they are released in specific amounts. They work in a clockwork sort of motion, which means they only carbohydrates is for carbohydrates, protease is for proteins. So yeah, like I said, so they're very specific. They have a very specific shape. So again, like I'm speeding through this, guys, very quickly. Can everyone in the chat define an enzyme? What is an enzyme? I want you to come up with a few criteria. 1 second, Rahul. I'll talk about that in a minute. So what is an enzyme? We know enzymes are... Thank you, that's the first one. A biological catalyst. Very good. They speed up the rate of reaction. That's true, Tarek, yep. Speed up rate of reaction. Now, did anyone say the last thing? Why is the last thing the thing that everyone forgets? Yo, George came in with it. Correct. Okay, without getting used up. Okay, so these are the three fundamentals of an enzyme in biology. In chemistry, there's actually another one, and that is by finding an alternate pathway, but we'll talk about that later. Okay, so in enzymes, biological catalysts speed up the rate of reaction without getting used up. You could also say they are specific to a particular substrate. Okay, and how are they specific for a particular substrate? Because of an active site. What's that? That's what a catalyst is, isn't? So why do you need to know the last one? I guess you're right. Yeah, I guess that's what yeah, you're right. You're right with that, Adam. I'll just put it in there just in case anyone forgets. Yeah. So we spoke about that. Oh yes, what is the definition of metabolism? I think that's a big question. What is the definition of metabolism? Sorry, I'm just looking at the stuff which people particularly get wrong. How much you're able to eat. I knew someone was going to say that. Well done, guys. Well done, Nathan. Well done, Manya. Well done, Yosma. If you did not know this definition, write this down. Metabolism is the sum of all the reactions happening in a cell or organism in which molecules are synthesized or broken down. So metabolism is the sum of all the reactions happening in a cell or organism. Okay? Josh, time and place, man. We're gonna go on a discussion, and I don't know if I'll see the end of it. In which molecules are synthesized. I can see that. In which molecules are synthesized or made into broken down. Guys, when they're broken down, are they soluble or insoluble? Are they soluble or insoluble? Just a question to see who's awake and alive. Very good. So these molecules are now soluble, which means they can be transported into the blood. That's the whole point of these things. Exactly. Okay. Right. We did that. We've done that. Don't need to know that. The lock and key model we already know, guys. So lock and key model just tells us that enzymes have a specific shape. So metabolism when people talk about nutrition, is kind of a different definition to metabolism when we talk about it in terms of chemistry. So this is kind of like a chemistry definition. This is not to do with nutrition and body and stuff. So my question was, does everyone remember what lock and key is, right? So lock and key just means the enzyme is highly specific in terms of a shape. It's highly specific. So essentially, what happens if it changes shape? Can it metabolize substrates anymore? Exactly, it can't. It just loses its ability and it can't do it. Just going to try and show you a diagram. By the way, is everyone okay with me kind of skipping over enzymes? I feel like it's something that you do in year 8 and year 9 and year 10. It just keeps coming up. It gets boring after a while. Just showing you what the lock and key looks like. Yes, so can you see the lock and key? So here we go. So enzyme, so substrate to this enzyme, it works. It's complementary. Guys, you do need to know these scientific terms. Did you guys hear what I said? You do need to know these scientific terms like complementary. Complementary means they fit like this. This is what they complement each other. They're a perfect fit. Okay, literally they work like lock and key. But here, they don't fit. Okay, so it won't metabolize. Right, moving forward, and here are the products. They go off broken up. So this could very easily have been glucose. Sorry, not glucose. This could have easily been starch, and then the enzyme metabolizes it, and now we've got a glucose molecule here, and we've got a glucose molecule here. And now the small intestine can do what? What can the small intestine now do? Correct, Ali, yeah. So what can digestion? That is digestion, by the way. This is digestion. Some could define digestion as this. This is digestion. This formation, this process here of turning large to small. That is digestion. So what can the small intestine do? Put it into the blood. Okay? So if this is the small intestine and this is the blood vessel, this is a large molecule. It turns into small molecules, and then the small molecules can then be transferred into the blood. Does that make sense? The small molecule can now be moved into the bloodstream. This is the blood. This is the small intestine. Guys, is this movement of molecules, is it passive or active? Is it passive or active? Very good. It is passive, which means what? It is going through diffusion. Now, what does the diffusion refer to? Passive means it's like, it doesn't require energy. Diffusion doesn't require energy. If someone does, like I said, a stink bomb in your class, they don't have to put in a lot of work to get that smell to start destroying your day. Whereas with active transport, there's a lot of work involved. So kind of, Tarek, kind of, you're thinking the right thing. So the molecules moving in, there needs to be a low concentration. And if there's a low concentration, the molecules will diffuse across. Okay? And therefore, this capillary needs to have a rich blood supply. Let me know if that makes sense. Oh, that's really... commiserations, Emmanuel. Rich blood supply. Let me know if this is making sense, people. Rich blood supply. So if there's a rich blood supply, that means there's a steep concentration gradient. If there's a steep concentration gradient, the molecules move out of the gut into the blood. Guys, I hope I have mentioned and done that justice. Let's move on. Now, the enzyme, is it a protein structure or is it made of something else? The enzyme, is it a protein structure? Is it made of protein? Did I make that up? Awesome. It is a protein structure, okay? So this protein structure has a very specific shape, okay? And also, we're going to talk about what affects its ability to do its job. Guys, just bear with me 1 second. Let me just put this here, just in case somebody's writing something. I need to do something real quick. Give me that 1 second. I think it's more like revision. I just want you to look at this and just like it's just a form of revision. Just a form of revision. Okay, moving on. Harris, how are we getting on? Let me know. Z as well, let me know. The effect of temperature on pH and pH on enzyme activity. So the specific shape of an enzyme is determined by the amino acids. So remember, shape is important. Agreed? Shape is important, okay? Because the specific enzymes are working with specific substrates. So the specific shape of the enzyme is determined by amino acids, okay? So the arrangement of DNA, therefore telling the amino acids to arrange itself specifically, will give it a specific shape. The three-dimensional shape of an enzyme is specifically important around the active site. So the active site, so if this is the enzyme, this thing here is the active site. Yeah, sure. Here, I'll put a highlight of. Okay? And so therefore it needs to fit into the active site. You said it, Adam, you said it. Enzymes work fastest at their optimum temperature. What is the optimum temperature in the body? 37 degrees. So at 37 degrees, the enzymes are working at 10-10 level. Okay, they're working really well. Exactly. So enzymes. I say 1 degree up and down, but even then, that can be a sign of an illness. Heating to high temperatures will start to break the bonds, okay, that hold the enzyme together. Now, you guys have learnt about DNA, right? Now, a DNA molecule, sorry, not DNA. You learnt about amino acids. Amino acids. What is the makeup of amino acids? Correct. What makes up amino acids, guys? Good. The usual suspects like carbon, hydrogen, nitrogen, all that stuff. Okay? So carbon, hydrogen, nitrogen. So they're made of elements, right? And these elements have bonds. So when the temperature gets so high, yeah, when the temperature gets so high, what happens? Those bonds break. Okay? The enzyme will start to distort and lose its shape. This reduces the effectiveness of the substrate binding to the active site. So I'll say that one more time. So imagine you have a long chain, okay? You have a long chain, okay? And that long chain has a specific shape. But when the temperature is too high, it's distorted, right? It loses its shape, it reduces its effectiveness, and therefore it doesn't allow a substrate to bind to the active site. Okay, so the active site is now gone. Agreed? If site is gone. So the active site is now gone. Eventually the shape of the active site is lost, and the enzyme is described as being denatured. So when I think of a denatured enzyme, essentially I'm thinking of an enzyme that's lost its what? Nature, I like that. An enzyme that's lost its shape. Yes, that's right, man. Yes. An enzyme lost its shape and therefore can't metabolize substrates. I'm moving on, people. Okay, not going to do that. I'll show you a quick diagram, really. I'll shrink it, I'll make it really small. Can you see the enzyme has changed shape? So small. So here's the amino acids, okay? All joined up together, making up the chain. The forces between the amino acids and the chain holding the enzymes together, it's unique. What happens? High temperature causes those amino acids to break bonds. When they break bonds, they lose the shape. You kind of have to remember that, yeah? So temperature change. High temp breaks bonds amongst amino acids and therefore loses specific shape. Handwriting is atrocious. I do apologize. I'm moving on, people. Let me know if that's okay. Okay, sure. Okay. So just a quick one. Why can temperature be good for enzyme activity? Why can temperature be... This is a question for everyone, yeah? Every single person. Why can temperature be good for enzyme activity? So let's see. Work faster, metabolize reactions. Guys, I'm looking for a physics word, you know. That's right, man. Yeah, I'm looking for a physics word. Watch out, everyone says the answer now. Why is temperature good for an enzyme? To an extent. It does work faster, so what physics word essentially goes in line with that? Nathan, man, you were cooking up a storm there. Well done. Man, yeah, as well. Good. Very good. So basically, if temperature is high, it means the enzyme has some kinetic energy, which means it has the ability to have more successful collisions. Does that ring a bell? More successful collisions. Guys, we are going to take a mini break now, yeah? I'm going to put this on the board so you guys can have a look at this. I think my break is even more warranted today because I've got the worst. You know what? That's a good thing. I'm glad to hear that. I'm glad to hear that. So I'm going to put this up for now. Guys, we're going to be back in 5 minutes. It is, but Ali, today I'm deteriorating. I'm like denatured at this point. I'll be back on... Nah, don't do that. I'll be back on top form for Sunday or next week. Guys, I'm going to be back. Did everyone hear that, yeah? We're going to have a quick 5-minute break. Yeah, we don't. Just now. I got 2 people that confirmed. I'm assuming everyone is silently confirming. BRB, people. BRB.